seeing us online. It is with pleasure that I open today's session of our SIG Pharmaco, a special interest group in pharmacology and therapeutics. I will be introducing this in English today because our session is going to be in English. So for the ones that are interested in translation, there is closed captioning in this platform that you can activate from now on and have the session translated into Spanish and Portuguese because these are the two languages that are active in the system. So this SIG Pharmaco, I will just briefly introduce our platform and the speaker of today's meeting. Is a monthly meeting platform coordinated by me. I'm Maria Cristina Velar, Department of Pharmacology, Federal University of São Paulo. With me in the coordination group, I have Dr. Roseli Godinho, that is also from my institution, Dr. Emiliano Barreto from Federal University of Alagoas, Dr. Paulo Guedini from Federal University of Goiás, and Dr. François Noel from Federal University of Rio de Janeiro. We are all the coordinators and the organizers of these monthly sessions. So the SIG Pharmaco is basically part of the telemedicine network. There is an initiative from the Ministry of Science and Technology and Innovation here in Brazil, supported by Studies and Projects Financer FINEP, and by Brazilian Association of University Hospitals, ABRA-RUI, and coordinated by the network of the National Institute of Education and Research Group, RNP, here in Brazil. So we are all very proud and happy that they are supporting our activities and giving us the chance to have these meetings that now we are in our 6 years of activity. So basically, the aim of this web conference structure is to reduce our geography distances and encourage integration and collaboration in teaching, research, and education in the area of pharmacology and therapeutics in Brazil. So currently, we are integrating here over 54 institutions that have graduate programs in pharmacology, graduate programs in areas related to pharmacology, and we also have participants that join us online through this initiative from the federal system. And so basically our audience is composed by students, researchers, teachers, but also professionals from the health area that are in hospitals, such as residents and physicians and nurses. So we are a very diverse and very interesting and good community with all interested in pharmacology. So I would like just to remind you for today's session. So for you that are here with us in live in this session, just to remind you that the microphones will be kept mute. I have already told you that you can use the closed captioning how can I say, o botão do closed caption para fazer a tradução para inglês, espanhol e português. And so we thank all the participants, and during the session at any time, please use the chat to express your comments, and then to include your questions that at the end of the session will be open for Dr. Page and Dr. Soraia Costa to respond. So we also remind you briefly of the new rule that we have. So you can record your participation and then be eligible for a certificate at the end of the session. So for those who have entered in this session using your login, or if you are new to the session and you use via cadastro, you have already put all your information, and at the end, you will see that there is an area where you can register your presence here And then later, the ones that want the certificate is just to follow the instructions. If you have any problem or question, please send us in the chat here or by email to the group. So just as an overview of our session, so after this brief introduction, I will be asking Dr. Soraia Costa. She is from the Department of Pharmacology in the State University of São Paulo. She will be the moderator of the session, and she will introduce our invited speaker. Next, our invited speaker will be presenting his talk. Dr. Clive Page will be the invited speaker today. And then after his presentation, we'll open the floor for questions and comments, as I said before. And after the end of the discussion session, questions and answers, I will be back here closing the session and give you the tip of who is going to be our next speaker and the theme of our next presentation in June. So with that said, I would like to, on behalf of the SIG Pharmaco Group, Dr. Page, to really thank you for your participation, for accepting this invitation. We really welcome here. And I also would like to express our gratitude to Soraia Costa, that was the one to invite you and to put you in contact with us. So both of us, our, thanks for the participation and collaboration with the group. So Soraia, I will close my camera now, and so I'll pass you the moderation of the session. Thank you.
Obrigada, Cristina. Thank you very much. And good morning, bom dia, colegas. I will give a brief introduction for my colleague Clive Page, because his CV is huge. I couldn't talk about his whole career today, but briefly, beforehand, I'd like to thank you, Clive Page, again. I know he's a very busy man, and to be here with us, and to be, he was yesterday practicing to try to enter in the system, because everything is in Portuguese, and I know it will be difficult for you to follow these steps to get access to this homepage or platform. So thank you very much again, Clive. So for my colleagues and friends, I'd like to thank you for the presence here. We have a good audience here, and then probably will increase by the time you start. So first, I'd like to say that Clive is a very famous man in pharmacology in Britain. He's British, he's married, he has 3 adult kids. I met one of them only. And he holds several prestigious titles and positions, including some that I couldn't translate what does mean. So he may tell us what does mean, OB, but he also has BSc, PhD. He has several titles, honor title for British Pharmacological Society. He's a professor of pharmacology at the Institute of Pharmaceutical Science, Faculty of Life Science at King's College. Clive obtained his BSc first class in pharmacology, and also his PhD from the University of London. He has been awarded more than 10 prizes during his career, and he is also a member of various associations, including British Pharmacological Society, where he served as the former president. He has secured over 100 grants for research and more than 10 grants for equipment, and he has supervised more than 50 PhD students, including some Brazilians. He's a member of the Executive Committee of the Federation of European Pharmacological Societies since 2022. He also holds several other external appointments. He has held or in some cases, he is still holding positions, including now he's on the board of our Brazilian journal. Clive has also acted as an expert witness to the FDA Advisory Board on Pulmonary Drugs, and he also appeared and continued to appear in several BBC TV and radio programs discussing about his major subject, drug development, and also asthma, and also very controversial topics such as research in animals, which is a big challenge in UK. He has served as a visiting professor in several countries, including in Brazil at Unicamp. Clive is also a member of the editorial board for more than a dozen scientific journals, and he also acts as an ad hoc reviewer for over 30 journals, including Nature, of course, BJP, Trends in Pharmacological Science, and so on. He has published more than 350 papers, over 200 reviews, I don't know how many book chapters, and he has edited more than 20 books and has written more than 20 editorials. So, Clive, do you ever sleep?
No, not very often.
So anyway, thank you very much, Clive, for joining us today. We are very honored to have you here. So it's all yours now. Thank you.
So thank you, Soraia, very much for the kind introduction. And as you say, I'm active in research, but I've also got a passion for teaching pharmacology and encouraging early career people into pharmacology. And really what I've tried to do, Soraia, for the group today is to talk to you about a particular course that I run called Drug Development Pharmacology. And this is a course that we run for part-time students as well as full-time students. And the part-time students are often people who are physicians or scientists or pharmacists who want to go into the pharmaceutical industry. And we have a lot of people also from regulatory authorities from the US, from Australia, from the UK. So it's a very mixed group. And we set this course up about 10 years ago with the Royal College of Physicians Faculty of Pharmaceutical Medicine, because they were concerned about how little pharmacology people knew that were going into drug discovery. So as you know, when you prepare for a new drug, you prepare an investigator's brochure, what is called an IMPD document. And even the reviewers that are reviewing these documents did not understand basic pharmacological principles, such as what is a PA2 to describe an antagonist. And so we put together a course with the faculty that now runs twice a year at King's, and we have people from more than 15 countries have been on this course over the last decade. So what I thought I'd do is just briefly take you through the timetable, because this module runs for 1 week and I teach for 2.5 days on this myself, all of the lectures. And as you'll see, we then bring in others. But as part of it, we really expect the students to be able to, at the end of the course, read an investigator's brochure, understand an investigator's brochure, and also what data they should be looking at, what questions they should ask when they're taking a drug from a preclinical setting for the first in man. And another module we run in parallel with this, which I can touch upon at the end. Actually, some of the students then have 1 week in a phase 1 unit, and they learn the principles of doing early experimental medicine. So I call this, Soraia, a BSc in a week. It's a science degree in a week. And my opening question to all the students is, out of 0 to 10, how good do you think your pharmacology is? And they say 5, 6, some of them might say 7 or 8. And then when they've told me where they come from, why they're on the course, I then ask them the difference between potency, efficacy, and affinity. At which point the students look at the ceiling because they don't know. And then we try and go through, you see on the first day that I have on this first slide here, we introduce them to pharmacological terms. So what is affinity? What is potency? What is efficacy? And really the whole concept going through the whole week is to what do you need to do to best understand the therapeutic window? Because Pharmacology 101 is about the therapeutic window. And we give them real examples. So these are not theoretical. where what sort of issues come up in preclinical work. For example, one of the species that we use for preclinical toxicology, the rat, cannot vomit. It can't show nausea. And yet it's one of the most common side effects in phase 1 clinical studies. And so we talk through how you might actually accurately assess and predict the therapeutic window before you go into people. And because many biotech companies and even big pharma often overestimate the therapeutic window, and therefore you potentially wait until you're having an early clinical study to find out some of the side effects. We then go through the basic fundamentals. And by the way, Soraia, I've shared my lectures that for all of these 2.5 days that I'm happy for you to share with your colleagues, because we don't have time to go at every lecture now. But we then do the fundamentals of discussing agonism, how you measure it, antagonists. And I think particularly with antagonists, because you can't measure potency, this is when we discuss PA2, why PA2 is important, how you measure PA2 if you're looking at an antagonist. And then we go into a lot of detail about inverse agonism, because there's a lot of, as you're aware, a lot of discussion now that many antagonists are no longer neutral, where they don't have an effect on their own, but they can actually act as inverse agonists. So this is quite a difficult concept for a lot of students to cope with, but I think it's really important. And also we talk about biased agonism, because there are now good examples coming along of biased agonists and what that means and where the drug development is going in this field. We talk also about the basic mechanisms of the different types of receptors that we have, the signaling involved, and why they're important physiologically, to understand G proteins, ion channels, the transcription factors, steroidal type nuclear receptors. So it's really a degree in a day, actually, to give all the basic fundamentals. And then, as you'll see at the end of the first day and throughout the week, with my colleague, Professor John Posner, he was the head of clinical pharmacology at Wellcome before it became part of Glaxo, we give the students really some data. We give them a draft investigator's brochure, and they spend the week in groups. And at the end of the week, they have to present whether or not they would take the drug into the clinic, and if not, why not? What other data would they be looking for? So this is sort of an ongoing series of workshops that the students, and they find this, so it's very practically orientated in the sense that we're hoping to give them information that's really of value of how to use pharmacology and what to look for. So then on the second day, I continue more of this, and we go into more detail about in vitro, particularly how you find drugs. So what sort of assays you use in vitro to find an enzyme inhibitor, to find an agonist, an antagonist. And we then start to talk about different body systems. And I start by talking about the autonomic nervous system, because if we think about the drugs that we have that influence the autonomic nervous system, we have so many different drugs for hypertension, for asthma, and it shows the principles of by understanding normal physiology, by manipulating it, you can predict not only drugs that will have a particular effect, but also their likely side effect profile. So it's a good grounding. And many of our concepts in pharmacology have come out of understanding sympathetic, parasympathetic neurotransmission. I then talk about my particular body system that I'm interested in, the respiratory system. And we also give them some other problems that relate to something they've heard from me about the skeletal neuromuscular junction, neurotransmission. And so we give them some problems about if you're trying to find a new nicotinic receptor antagonist or neuromuscular blocker, what things would you look for? What models could you use? So it's more of a practical approach to using some of the information they've learned during the week. And then the rest of the week is, as it says here, we try to cover different disease areas, the CNS, cardiovascular, and I bring in people externally to the course, but are within King's, who are involved in drug development in these different therapeutic areas, to talk about both the challenges, what animal models there are, whether they're good, whether they're not, and also where some of the areas are interesting of new pharmacology coming along, new approaches. So by the end of the week, we don't cover absolutely everything, but they get a feel for, based on the principles we've taught them in the first day and a half, how you apply that and what specific problems there are in developing drugs for different therapeutic areas. So you can see here, we talk about schizophrenia. We have people talking about failures from using models to predict, for example, ischemic heart disease. And we also have a number of self-assessment exercises. At the end of each day, the students are given a series of questions, they're not assessed on this. It's more, have they learned anything from us, what they've not learned. So as we're going through, and we use this as feedback to try and improve the course as we're going forward. And then on the last day, they do workshop presentations. We talk about cancer, which is a very big area. We have a lot of research going on at King's in this area, and a big interest of mine. And we have a strategic collaboration at King's with our Public Health England, where they are developing new anti-infective drugs, antifungal, antimicrobial. So they see a lot of this, and then there is some feedback. So that's the sort of overall course for the week, but there are parallel courses they can choose on pharmacokinetics, phase 1 studies, pharmacoepidemiology, but they're different weeks in the year, and they build these up to then form for a master's degree in pharmacology or clinical pharmacology. So then if I can have the second presentation I sent across, I don't know if Emiliano is there or not. Yes. So what I thought I'd do is just take you through a particular drug development program that I've been heavily involved with. The drug that I'm going to tell you about is now with the FDA, and on June the 26th, we hope they will approve this drug. But I use this as an example of how you go from an idea that we had within the university and take this through, and the different stages that you go through to take a drug into man for the first time, and then moving forward to do clinical trials, in this case for asthma and COPD. And I should acknowledge Dom Spina, my late colleague who sadly died a few years ago. And for those of you who know Yanira, this was Yanira's husband, and who worked very closely with me on this project. And just to say that one of our undergraduate students, the first author here, actually did a placement project in my laboratory where she did the first experiments on this drug, just to show you that academics can find new medicines and take them all the way to the clinic. And then to acknowledge David Jack. And for those of you who don't know David, he was one of the leading pharmacologists. He was the research director at Glaxo in his previous career. And he discovered salbutamol, salmeterol, ondansetron, sumatriptan, and he also discovered ranitidine. So he was a really incredibly good pharmacologist, and I've learned much about my translational approach to pharmacology from David. So the first thing we do, and I think this is true for any project in drug development, is actually to say, well, what is wrong with the drugs that we have? And in this case, for asthma and COPD, we have some good drugs, but why do we need new ones? Because if you can't justify that and getting across to the students that you really need to have a clear understanding of why you're developing a new drug, and also where it will likely fit in. So we try and get the students to understand the so-called TPP, which is really what does the product profile at the end look like for your drug if you're successful. Because if you can't see where this will fit in and add value, you shouldn't start the project. So we do this for any drug class that we're looking at, but this is just happens to be used for asthma and COPD, where steroids are extremely good anti-inflammatory drugs, but they have side effects. And the world would like to have a drug that is anti-inflammatory and not steroidal. And so we then go through a little bit of history. So this drug I'm going to talk about is a phosphodiesterase inhibitor, and I haven't got time to go into all of this, but we've known, of course, that with xanthines, we've had non-selective phosphodiesterase inhibitors. I take them through the recent understanding of the different molecular classes of phosphodiesterase there are. And we also talked to them then about where some PD inhibitors have already been developed, what they've been used for, what some of the side effects are around this. So obviously around PD5 inhibitors for erectile dysfunction, pulmonary hypertension, but particularly around in this area, the discovery of phosphodiesterase 4 inhibitors, a number of which have been approved not only for asthma, but also for psoriatic arthritis, and some of the issues around side effects of these and how you can improve on this, because if you can't improve on it, you shouldn't start the project. So we then go into a little bit about why phosphodiesterase are important. And again, I don't need to cover this with you today, but just really so they understand that there's a rationale for targeting phosphodiesterases in the context of targeting inflammatory cells and airway smooth muscle. We also talk about the presence of these different enzymes in different cells, so they have a good background to that. And then really the unique thing about this drug that I've helped develop, and it's called RPL554, it's now called ensifentrine, was that it was a bifunctional drug that inhibited both phosphodiesterase 3, found in airway smooth muscle, and when you inhibit that, you get bronchodilation, and phosphodiesterase 4, which is the predominant cell in inflammatory cells. And we already know with roflumilast, if you inhibit this, you have a drug that's anti-inflammatory. And so we were trying to find a drug that would have broad approach to treating asthma and COPD by having two really important pharmacological properties in a single molecule. And the question is, where do you start and how do you end up with a drug like this? So from David, we worked very closely because the challenge was, how do you get a drug with two activities to make sure that the activities have the right proportion of inhibiting 3 for bronchodilation, inhibiting 4 to block inflammation. And very important is that we wanted this drug to be at least twice daily active. And so it needed to have a good duration of action. As I'm going to show you, despite all the reductionist approaches in molecular biology that are being used in drug discovery, we used very much a phenotypic approach because we wanted to know early on whether we have a good duration of action. And that was a critical point in the project. So one of the things those of you who know anything about phosphodiesterases will know, there have been many drugs developed that have failed because they cause nausea and vomiting. And so we were very clear we wanted to start with a chemical pharmacophore that wasn't actually related to roloperam, which was one of the earliest PD4 inhibitors. And we came across this molecule called trequensin. And we chose this drug because it had been developed to treat the cardiovascular disease by a German company called Herxt. It had been into human beings and failed. It had no cardiovascular effects whatsoever. It caused no nausea and vomiting, but we identified it was a PD inhibitor. So we started to make analogues of this particular compound because we knew that the mother molecule was not actually emetic, and that was a very important consideration. And so the very first experiment we did, and David was the person who suggested this, and for those of you more classic pharmacologists, and I know lots of you in Brazil are still true classical pharmacologists, we did a very simple preparation of taking the guinea pig trachea, electrically stimulated, and get these beautiful contractions, which we know are parasympathetic, this acetylcholine. And you'll see here that the same tissue is maintained for 6 hours. So we had a very nice basal assay. And this is really key to have an assay that is robust and reliable for your early preclinical work. And then we could do with trequensin itself, we could show that when you put the drug onto the tissue, you got a nice concentration dependent relaxation. And most importantly, this was quite a slow recovery time. So not only was trequensin a drug that had been in people and had a good safety profile, but it had exactly what we were looking for of a good duration of action. And this again was absolutely critical part of the project because we needed a long-acting drug. And so from that, we went and did actually later to show that the drugs that we were looking at blocked recombinant phosphodiesterases. So we can buy all of the recombinant enzymes 1 through 11. We looked at functional assays, both in vitro, to show that not only, as I've just shown you, the drugs would relax and have a good duration of action, but could suppress inflammatory responses, because they were the two pharmacological effects that we wanted in the final molecule. And again, this is the primary screen. So if you talk to any biotech company now, they'll tell you their primary scheme is putting a receptor or an ion channel on a chip and having robots screen thousands of molecules. We did not do that. And we did this quite deliberately because we wanted to know, more importantly, that the molecule relaxed the tissue rather than whether it was very effective at a particular enzyme, at least initially. And so we only screened 182 molecules, which we had made externally by a CRO. And you can see just a couple of examples here that early on where the reversibility was quite quick. They didn't have the right duration of action, but nonetheless, they were very effective. And so we could do structure activity relationship around these molecules to find something that was longer acting. And we ended up with this molecule, which now, as I said, is RPL554. And you can see that very quickly it caused a very rapid relaxation of airway muscle with a fantastic duration of action. So it's exactly what we wanted. And so with this, we then had to raise some money to be able to develop the drug further. And of course, to do that, you need to know a lot about its appearance, how stable it is, all the things that are needed with any new chemical entity, the pKa, what it's soluble in, and we were going to give this by inhalation. So would it be compatible with different excipients that you could give by nebulization? All of the classic stuff. And all of this we've done effectively working with third parties because we were a small academic group. But it highlights to the students, again, the information you need to have when you go to a regulator that you have in your investigator's brochure. And this is the sort of information we give to them. And you might have a compound, for example, that is racemic. Have they considered the enantiomers? Do both enantiomers behave the same way? Do they have different pharmacology? Because often they develop a racemic compound and don't fully understand what the different enantiomers do. So coming back to this nice assay here, and again, this was an undergraduate student of ours who set this assay up, and you can see this beautiful contraction. The moment, the first ever experiment she did with the drug, you can see you got this amazing suppression of the contraction of the trachea. But of course, this is in vitro. We had to repeat this multiple times, and we had to show this was real and not due to any solvent or anything that we were doing. So good experimental design. We tried to build in teaching our students as well. And we again showed it was a dose or a concentration dependent relaxation. You can see a very, very clear persistent duration of action, despite it's not always giving a maximal effect depending on the concentration you use. And then we moved in vivo, because this is something I know, Soraia, you asked me to talk about, is really that we were very much focused on phenotypic responses, because as David said, if your drug doesn't relax tissue, I don't care what the mechanism of action is, it isn't going to work. And you show first that it relaxes, and then you understand more about what it does. But more importantly, can you deliver the drug to the lungs safely at a dose that gives you suppression of bronchoconstriction in vivo, because ultimately that's where the drug's going. And so this is just an early experiment that Dom Spina did with me, where we took lactose powder. You can see here in an anesthetized guinea pig, and I'd like to say as an aside here, this drug has never seen a mouse, and we can come back to discussing mouse experiments later. The guinea pig is by far the best preclinical species, in my opinion, for respiratory drugs. But you can see when we give increasing doses of histamine, we can measure airways resistance to look at airways obstruction. And you can see over a period of 5 or 6 hours, this is actually a stable in vivo preparation. When we give a drug in the dry powder in lactose, you can see that you get complete suppression or near complete suppression. So we knew the drug was able to also bronchodilate in vivo. And we could show a long duration of action. And we, in parallel, then started to look at, well, we thought this was working through PDE. And you'll see here that it's a much better PD3 inhibitor than a PD4 inhibitor. The IC50s here against the enzymes are actually quite different, but they're very, very selective compared to other of the known PDEs. And so we wanted to really understand, if it wasn't such a good PDE4 inhibitor, would it nonetheless still inhibit inflammatory cell activation? Because that was something that we were obviously very keen to do as part of this bifunctional drug. And so in parallel to these in vitro experiments, we also very quickly did some initial screening to make sure the drug hadn't any acute toxicity. We looked at all sorts of other profiling against other targets, so off targets, to make sure. And again, this is very important for the students to understand that the earlier you find out about any safety concerns, the better, because then you can try and mitigate them. And we started doing safety pharmacology. Does it affect the CNS? Does it affect behavior, etc.? Because all of this you need as part of your investigator's brochure. And we looked primarily at the respiratory and cardiovascular systems. We did this in 4 different species. And I may have time to show you, we also did some functional in vivo work in primates to show what we'd seen in guinea pigs translated, because the primate is obviously closer to man than guinea pigs, but they are expensive and we don't do experiments in primates lightly. So in fact, here is the experiments we did. So these are animals that are naturally sensitive to Ascaris suum. And you can see that when you give the drug into the lung, you can prevent bronchoconstriction both to methacholine, which is used clinically as a marker for airway responsiveness in patients, but it also blocked the antigen-induced bronchoconstriction. So you can see what we're doing here is taking the students through the actual development program, why we chose the species we did, why we chose the experimental setup that we did. And then we also, and very importantly, coming back to the therapeutic window, measuring heart rate, measuring blood pressure in the same animals that we're looking at bronchoconstriction. You can see at the same dose, there's no effect where we're affecting the airways, there's no effect on blood pressure or heart rate. And that was a really key experiment for us to making a decision alongside all the other safety pharmacology that we felt comfortable to go into people that the drug would actually have an effect and actually have a reasonable safety profile. And remember, the parent molecule for this drug had been into patients previously and shown no cardiovascular liability. So this really confirmed that this analog also had no problem with the cardiovascular system. And just the final bit of the preclinical work, we obviously needed to show that the drug was anti-inflammatory, and this is just a typical model of allergen-induced eosinophil infiltration, which many of your colleagues in Brazil use similar models. And against a standard glucocorticosteroid, fluticasone, and the phosphodiesterase that's clinically approved, roflumilast, you can see it gave a very good anti-inflammatory effect at the same dose that we used for obtaining bronchodilation. So there are other things you have to do. Although it's by inhalation, there are CYP enzymes in the lungs. So we needed to understand, did it interfere with CYP? Because this would be a drug given with other medicines, particularly in patients with COPD. We did PK studies by both intravenous and inhalation to show that there is the bioavailability, which is very, very low. We had to develop a new assay to find this drug because it's so poorly bioavailable, which is exactly what you want with an inhaled medicine. And we obviously looked at cytotoxicity, phototoxicity. I talked to the students about where we went to the regulators early for scientific advice, and they said, well, your compound's slightly yellow, you should do some phototoxicity. And really to get them into this way of thinking that you need to talk to the regulators and see them as your friends, not as your enemies. And then there's a whole list of things I haven't got time, but we went through 2 28-day toxicology studies in both rats and dogs. And we also found a small effect on hERG, but at a concentration that was way in excess of anything that we could achieve and found in plasma of any species we looked at. Again, we sought advice from the regulator. And although it works at this very high concentration, we now know in the clinic in more than 3,000 people, there is absolutely no effect on QT interval with this drug. And so, but this early safety pharmacology, was that if we'd have found something against HERG at a concentration that was likely to be needed to get a biological effect, we would have stopped the project. And my advice to all students is you look for ways to stop your project, not for ways to keep your project going, because if there's a problem, you need to identify it early and not waste time, money, and animals for something that may actually have to be stopped later on. Now, you can never predict, but it's a good philosophy is kill quick. And so many biotechs don't do that. And I think as a result, waste a lot of time. So again, there was no genotoxicity, mutagenicity. We talk about why we do this. We also look about predictability for carcinogens, because obviously you don't do long-term carcinogenic studies until later when the drug's been in more people. And then just go through what we observe, but how you design the 28-day tox, what that allows you to do before you then press the button to go into human beings. And we sit down and discuss at that point, well, what would you do and how would you take it forward? And here's the first clinical study we did in the University of Leiden in Holland, showing you that the very first person who had this drug, that we measure their FEV1, gave a 16% increase in FEV1, which is a profound, it's as big as salbutamol compared to placebo. So exactly what we'd seen in guinea pigs and primates, now translated into patients with asthma. And then we take them through, we did an early study in COPD patients because they are patients who have cardiovascular comorbidities. We wanted to check the safety, particularly that this was not going to cause problems in patients with cardiovascular comorbidity. And then we also proved in patients that it was anti-inflammatory. And this is a study where we gave lipopolysaccharide to humans. We did an analysis of their sputum and showed that the drug had an inhibition of the neutrophils coming into the lung. And then we showed some preclinical work with human airway smooth muscle, and much of this is now published, where we looked at interaction with known bronchodilators and showed a profound synergy with muscarinic receptor antagonists and beta agonists, which are the two primary classes of drug used to treat respiratory disease. This has now been also confirmed in the clinic and is going to be used on top of other drugs. So this was an important preclinical experiment, and you can see an early clinical trial here showing the profound potentiation of adding the drug to tiotropium, the muscarinic receptor antagonist, compared to just tiotropium alone. Now, for those of you coming later this year to your Brazilian annual meeting where I'm going to be speaking, we can talk about this in more detail. I'm rushing through it now because of time, just to show this is an example of drug development that I've been involved in that you can share with the students and help them understand. And so there's just a summary here of really what the advantages are, what side effects are, why it's worth spending the money to do a phase 3 trial. And I won't go into it now, but we had to raise $200 million. We then did 2 phase 3 trials, which were successful and have recently been published. And we hope the FDA think that the package is good enough. But it's what I've shown you is the early preclinical work, the way that we use phenotypic screening, the decision-making points in this project that allowed us to go into man for the first time as a real project to get them to think about it, all the way from why do we need a new drug to when you would stop a drug, and indeed what sort of further data you might need going forward. And I just finished at this point by showing you that we published the first paper of all the clinical studies in the Lancet Respiratory Medicine back in 2013, which were 4 clinical trials that we did showing that the efficacy and most importantly the safety of this drug, it was a new chemical entity, and really that this is a drug that we hope will find as a new first-in-class drug with bifunctional activity for treating respiratory disease. So my colleague John Posner, as I said on the timetable earlier, he does a similar thing with them with a new anti-platelet drug that he's worked on, and they also do something on the neuromuscular blockers. So over the week, they get exposed to 3 completely different drug development programs, but we go through the same process. So I think, Soraia, I've spoken for about 35, 40 minutes. I could go on for longer, but maybe it's better that I stop now and we have a chance to discuss anything that comes up about the course, about the drug, about how we approach this. Rather than me keep talking, I think it would be good to have the rest of the time for discussion if you're happy with that, Soraia.
I think you're fine, Clive. If you want to carry on the in vitro talk, if you wish, because we have.
Yeah, okay. If you want to put up the in vitro one as well, I can certainly touch on that too. If you put up, there's one called Fundamentals, I think, Soraia. I can talk a little bit about that if that helps.
Yes, Emiliano, please, can you carry on?
Emiliano, can you help?
I try.
Yeah, I think there's one Emiliano, not this one. It's called, I think it's called Fundamental, or this can do. Yeah, we can start with this one if you want, Soraia. So this is on the first day. If you can give me there we go. We take the students, as I say, the whole purpose of the course is at the end, they can look at an investigator's brochure and they see all these measurements for affinity, for potency, whether it's an agonist, an antagonist, and they have no clue what they mean. And so we try and talk to them about how you measure these things, why they're useful, and how then you can use it as part of your assessment for evaluating the drug going forward. So, and I worked very closely with the head of preclinical pharmacology from Pfizer before it should shut down in the UK, where we went through what you're actually doing as part of a screening sequence. So what are you trying to do? You're trying to understand whether your drug is selective for its target, whatever that is, its duration of action, as I talked about with our drug, all the off-target potential receptors and ion channels that you obviously don't want to hit, but you need to understand if your drug at the same concentration hits a sodium channel, may it have local anesthetic activity that you don't want. We talk about different tests in vivo that you would use really to start beginning to understand the therapeutic window. And I also, Soraia, talked to them about the idea that there's no single number for the therapeutic window, because you often see people say the therapeutic window is 10 or 20, and if it's less than that, we're not going to take the project. And I use the example of salbutamol. The therapeutic window for salbutamol intravenously is very different to when you inhale it, which is why we inhale it, to keep it away from the blood. And this is something they don't really think about, but actually is why we choose the routes of administration we do, and it's often a way of trying to widen the therapeutic window and giving some real examples of where that happens. And then, of course, you need to understand bioavailability, which with an inhaled drug, you hope is low. But for your oral drug is sometimes you have a fantastic drug in a test tube, very potent against a particular enzyme or receptor, but actually is so badly absorbed that you'll never get a plasma level. So we also talk about that there's more now than the classic pharmacology of agonists, antagonists, and our concept of antagonism is really around neutral antagonists that bind a receptor but produce no effect. They just block the effect of the agonist. And that now modern pharmacology is challenging these concepts because we have all sorts of other things like biased agonists and inverse agonists. So we talked to them a little bit about, and I don't go into the maths too much with this because I think it's not important for them to know all of this, more about that you can use what assays you can use to identify drugs. You can look at their off rate, their on rate, which for antagonists particularly, you may want a slow off rate so that you get a good duration of action. Some of the challenges with the different types of assays that are done to look at the antagonists particularly. And then we go through this where just looking at affinity to a receptor doesn't tell you whether it's an agonist or an antagonist, and it certainly doesn't tell you very much about it. You have to build in some functional component where if it's an agonist, you can actually look for efficacy and potency by measuring, for example, in a test tube, a G protein or a calcium signal. and then in vivo, where you can use some functional readout, as I showed you, like bronchoconstriction, where you can estimate the dose that can give you a 50% inhibition of the desired biological effect. And again, so we go through some of these terms, and you know better than I, and how you measure some of these things, the off rate, the on rate, and really so they get a good understanding of what these things mean, that we don't put them into an investigator's brochure because we like putting numbers in. They're used for a particular purpose. And I think particularly important is understanding, particularly for antagonists, how many receptors do you need to occupy to get good inhibition? And if you look at this, you know, what is the concentration required to give more than 75% of the receptors being occupied? Because if you can never achieve this concentration safely in your in vivo experiments and ultimately in man, the drug's doomed to failure. And therefore, by understanding this early, you get a good understanding with an antagonist whether or not you can ever achieve a concentration that can act to occupy and block enough receptors. And this is something, again, that by using real examples, they I think get at the end of it. So we talk about agonists, how you measure relationship, partial agonists, you know, whether the difference between agonists of different potency, and some different concepts. So if you look here, this is a real example of developing the long-acting beta-2 agonist indacaterol from Novartis, where they actually looked initially in sort of an in vitro system of binding. So does the drug actually recognize the beta receptor? Then a functional assay in vitro looking at cyclic AMP elevation as a functional readout. Then they look at the tracheal assay I showed you of the contraction of the trachea, and then very importantly doing some human tissue. And the importance of showing them this slide is that whilst the absolute numbers vary across the assays, the rank order of the agonist actually is absolutely the same all the way across. So formoterol is always more active than salmeterol, than salbutamol, whatever the assay, so that you know you've got a drug that is behaving as an agonist, and even in human tissue, which gives you confidence before you go and do clinical trials. Again, I won't go through all of this for time, but there's a lot of stuff that we also talk about. And with antagonists particularly, is really just looking at, you can't measure directly an effect of a neutral antagonist. It has no effect in a cell. So we go through some of these examples where you look at how good or how much of your antagonist you need to occupy 70% of the receptors. You look at the plasma concentrations, and you can see why some drugs work and some don't, because they go into the clinical trial with concentrations they can give safely that are not enough to occupy more than 70% of receptors. And I think this is something they then begin to understand why you need something like the PA2, if I can move on and find it, but really why we have the Schultz analysis in terms of, to be able to look at an antagonist, you have to have an agonist and see how effective it is at translating or inhibiting that agonist response. And this is something that they've been taught it often as undergraduates in a very different way. They just see all the numbers and equations and don't like it. But when you explain it absolutely in the context of, is this a high enough concentration from the Schultz plot that you can achieve safely clinically? If it isn't, don't start your project. And you can look at this information in the investigator's brochure, but they just look at the number PA2 and don't really understand why someone's looked at it. So we're really trying to boost their confidence to look at the data in front of them, ask the right questions of the drug that's in development, and therefore would they make the decision to go very forward into man. So I don't know, Soraia, whether that's enough. You want to go on a bit more, but we use real examples to show them about how you modify drugs to give you a longer duration of action, exactly as I showed you with my example. But we really try and get them to think. So we're not asking them to remember equations, but really have they at the confidence at the end of the week, Soraia, to pick up a brochure of a new drug, look at it and say, would I make the decision to go into human beings for the first time or not? And if not, what's missing and what questions could I ask? And things like chirality I mentioned is really key. So I'll stop there. I could go on. Remember, this, what I've talked to you takes 2.5 days. So I apologize if I've gone very quickly, but you get a flavor of what we try to do and how we try to illustrate this.
Fantastic, Clive. I think we had a taste of your course, and of course, our aim now is to bring you here for longer, in addition to the meeting, of course, next time.
Yeah.
But,
well, it's sunny in Brazil, so maybe I should move to Brazil, Soraia, because it's very miserable at the moment in Europe.
You have a lovely garden, but I think if you don't have sunshine, it's difficult, isn't it, to be there?
Yeah.
Yes, yeah, you are welcome. I think the pharmacologist is going to be, we're going to be very happy to have you here, because this course is amazing. You just go for the basic to very complex, and I may need to do this course as well to remind me some.
Yeah, well, I think that's important, Soraia, because a lot of the students at the beginning, you say, I'm good at pharmacology, I've got 7. And I said, well, why are you on the course? But at the end of it, they say, we now understand pharmacology because it's not theoretical. We really try and instill in what sort of information they need, because it's around drug discovery and development. It's not theoretical. So that's key to it.
Very good. I think you should do a book only on this subject. Just very, very nice. Anyway.
So I noticed there are some questions. Sorry, I don't know how you want to, but do you want me to answer the ones I can see? Do you want to have people?
Yes, go ahead. You have some from Marco, and then hope more people can. Yes, go ahead.
So hi, Marco. So yes, we have looked at airway hyperresponsiveness. The drug's actually being approved, we hope, by the FDA for COPD initially, but I showed you the very first clinical experiments were in asthmatics, and you can shift the methacholine dose response curve very significantly. And preclinically, when we give allergen to the primates and the guinea pigs, you can inhibit hyperresponsiveness, not just inflammation. So, and that's not just because it's a bronchodilator, it's quite a long-lasting effect on hyperresponsiveness. Marco also asked about, is it effective in glucocorticoid resistance? So we don't know the answer to that. It's been done on top of patients who've been given dual bronchodilation, in some cases also steroids, and showing an extra benefit in terms of reducing exacerbations clinically and improving lung function. But we've not specifically looked at patients who are resistant to steroids. That may come down the line, and I'm no longer involved with the development of this drug. When it went to the phase 3, it got handed over to a company who've got the money to do that. So, but it's a good question, because it's not just about finding an alternative to steroid, but can we actually reduce steroids or when people are resistant? And this is particularly true in COPD, because we now recognize a lot of patients taking steroids with COPD have an increased pneumonia risk, because you hit them hard with a steroid, the bacterial levels go up, not down, because you've hit host defense. So I think, whoever finds an alternative to a steroid, it will find great use across a number of inflammatory diseases, not just respiratory. The next ones are, someone's asked the question, I was wondering, it's a big deal leading with polymorphism and drug development.
Sorry, just 1 second, Professor Page. I'll ask you or Soraia to read the question, because then it will be easier when people are watching on YouTube to follow the answer with the question. Okay, thank you.
Yeah, well, I read out the Marco's ones, but so the question from Douglas D'Souza is, I was wondering if it's a big deal leading with polymorphism receptors and drug development. It might impact in those steps you were talking about. So you're right. I mean, one of the things that we were concerned about, Douglas, with this project was that there are 11 different phosphodiesterase enzymes, and we only wanted to hit 2 of them. But within phosphodiesterase 4, there are actually something like 24 different isoforms. and whether some of them are more important for side effects than others, which people have suggested. And if we think about the beta-2 receptor that I touched on here on the slide we've got, we now know there are lots of polymorphisms in the beta-2 receptor. Many of them don't make any functional difference to whether you respond, but there are a subgroup of people that do not respond very well to beta agonists and actually sometimes can have an adverse event. And it's now recognized that some of these polymorphisms may predict that. So it is definitely something that you need to consider if you know there are polymorphisms in your target. We were just lucky with the ones that we've looked at. We've not really had an issue that we've had to deal with. What next? There are some of these in Portuguese, Soraia. I don't know if you can see them. Maybe you can read them out and I'll try and answer them.
Yes, it was only me asking people to send their questions, but
okay.
one for you as well.
So there's another one here then. Given your extensive experience in pharmacology and drug development, what do you believe are the most significant challenges currently facing the development of new drugs? I think the biggest challenge, Soraia, is that we are developing drugs that are often for, in the past, diseases that are common, because industry wanted to have a drug for hypertension and everybody take it. We now recognize with personalized medicine, and we've just talked about the different receptor subtypes that may exist between person A and B, is really trying to be more personalized in the drugs that we have, that, you know, we aren't all the same and we shouldn't therefore all have the same treatment. And I think that is a real challenge, but there's a lot of talk about it. And outside of cancer, I think it's quite difficult to show concrete examples where someone has gone for a particular indication for that subgroup and it's better than giving everybody a drug. Now, with cancer, there is obviously markers that change in a particular type of cancer in some people and not others, and you can target that, and it's not normally being expressed in the same way, which is not always the case. And the question that came up about host defense really is a real problem for us in the anti-inflammatory area, Soraia, because any drug that suppresses lymphocytes or macrophages, they are part of homeostasis and host defense. And if we hit them too hard, we have a problem. So we need to start looking for pathways that are upregulated in a given disease, so we can only knock out the bit that's relevant to the disease. And that is coming. And I think it's a really interesting challenge. And I think you'll start to see more drugs along that line. But at the moment, we do still have a lot of drugs that I think are being targeted for everybody with a particular indication. Now, one good example that gives me hope in this area is what's happened in cystic fibrosis, because you've now got these ion channel modulators that really restore, not quite cure people who've got a particular genetic mutation. The problem we're left with is there's a very small number of what is already a rare disease that don't have that mutation and don't respond to these ion channel modulators. And who's going to find the drugs for them? Because they've now got to see their contemporaries with cystic fibrosis doing fantastically well. The other big challenge we've got is around anti-infectives. You know, just a few weeks ago in Nigeria, it was published in Nature, we have the first patient resistant to colistin. We don't have any antibiotics after colistin. If you become resistant, where are the new antibiotics coming from? So one of the other projects I'm currently working on with colleagues of mine here is to try and find drugs that aren't antimicrobial, but can restore the sensitivity to existing classes of antibiotics. Because if you develop a new antibiotic this week and it's approved, Soraia, your government, my government, the FDA, the American government will put it in a drawer to say it can only be used in patients when everything else has failed. So there's no commercial market and therefore no incentive to develop new drug classes. And that's something that WHO, our government should be working together to incentivize people to develop antibiotics. And even if they're put in the drawer, you actually cover the cost of the development, which, as you know, is anything between 800 million and $1 billion now to find a new medicine.
Great. Thank you, Clive. There are two questions from Michelle Pereira. She said, yes, excellent conference, Professor. Thank you. Good morning. The routes of administration of the inhibitors that the professor studies, is it inhaled or oral? During the test, carry out tests with both routes?
Correct. So the drug has gone into the clinic by inhalation. And we did this deliberately because we wanted to keep the drug out of blood, because the previous PDE4 inhibitors affected nausea and vomiting. And also PD3 inhibitors can cause problems with congestive heart failure and vasodilation and falls in blood pressure. But as part of the preclinical assessment, we did oral dosing, we did intravenous dosing, and we did inhalation, both for looking at pharmacokinetics. But people forget that even when you inhale a drug, depending on the device you put the drug in, up to 90% of that drug is swallowed. So if it's oral inhalation, because some of that oral drug, if it's bioavailable, will get in, and you can just have as many side effects as if you were taking a tablet. So you need to understand what happens with oral dosing, even with an inhaled drug. Now, some of the newer inhaled glucocorticosteroids, which are inhaled again to keep them out of blood, they have this secondary defense mechanism that the swallowed portion goes through first pass metabolism, so that what enters the circulation is then negligible, and that's why they have a much better safety profile. But you have to really look at all routes of administration to understand the fate of your drug. And even though you're trying to keep it in the lung, if 90% is swallowed, you need to understand what happens with that too.
Great, thank you. So Christina now is asking, first of all, she's thanking you for the excellent presentation, and she's asking if PD family members are presenting gonads, any association of your compound analog with the effects on the endocrine system, for example, testes and androgen production?
Yeah, so it's a really good question, because there are certainly with phosphodiesterase 4 effects on the endocrine system. But again, it's partly why we chose the inhaled route, because that way we had, as I said in the talk, we had to find really a new assay to estimate how much drug got into the plasma, because it was so low. So the amount of drug that got to the heart, that got to the endocrine system, that got to the gonads was negligible. So from the 28-day toxicity testing, we saw no effects on the endocrine system. The dose limiting effect, which is very, very, very high, was actually on heart rate. And the heart rate we saw was less than you'd see with a conventional dose of salbutamol. So you have to give a really large amount of this drug to get into blood to have any systemic effects. But the question is right, and other people are trying to find PD4 inhibitors to manipulate the endocrine system for good. But with this compound, we were trying to keep it away.
Good. Marcelo Muscara, my colleague, said your talk was very nice. Thank you. Am I wrong, or the main challenge in targeting different pathways with a single compound is having similar affinities for both targets, which was the case with the RPL compound on PD3 and PD4? Could it be different? For example, dimerizing different groups of drugs like beta-2 agonist plus corticosteroid hybrid.
So a really, really good question also. So I showed you the in vitro data, that it's much better in vitro at inhibiting PD3 than 4. And if we'd have started with doing the in vitro assay, most companies and most people would have said, it's not bifunctional, don't start. But I used a phenotypic assay, and we showed it was bronchodilator and anti-inflammatory at doses you could give safely. And it was only later on we went back to look at, was it a PD3/4 inhibitor? Because in the functional in vitro assays, and I didn't show you this, but if you take human neutrophils or human macrophages and you stimulate them with LPS, we know that that's susceptible to PD4 inhibitors. And this drug is very effective at switching off TNF alpha release, for example, whereas PD3 inhibitors don't. So even though this drug is a good PDE3 inhibitor, the 4 component was good enough to inhibit inflammatory cells function, and that's what you really want. The other thing, of course, is that in many cells, including inflammatory cells and airway smooth muscle, we have 3 and 4 in the same cell. They're compartmentalized, and there's previous data that shows that when you give a PDE3/4, a 3 inhibitor and a 4 inhibitor together as 2 separate molecules, you get a greater effect than if you give a single inhibitor of 3 or 4. So we forget these enzymes are actually talking to each other and cooperating. And when you inhibit both, I think you get effects that are greater than hitting one enzyme alone. So your second question about dimerizing different groups, you're absolutely correct, and people have done this. So there's one group in China that has taken roflumilast as a PDE4 inhibitor, and they've covalently linked it to a beta agonist. So they're trying to get the bronchodilator anti-inflammatory in a single molecule. There are other companies who've taken a different approach of having a beta agonist on one end and a muscarinic receptor antagonist on the other end. And these are called MABAs. And whether they will do anything different to giving the 2 drugs individually, we don't know. And the challenge is you have an antagonist on one end and an agonist on the other. What do you dose it on, as the antagonist or the agonist? Because the antagonist, you want to stick on the tissue and stay there. The agonist probably goes on, has an effect, and comes off. So the challenges there have been, including some of these being in phase 2, is how you dose it and whether it will give anything better than putting a beta agonist and a muscarinic receptor antagonist in the same inhaler. But there are people taking this approach. And I think, as we've seen in AIDS, as we've seen in cancer, we've seen in tuberculosis for more than 50 years, no single mediator is going to be the answer. So the more that we can broadly target multiple pathways, but this, of course, is against the backdrop of the last 20 years of the molecular revolution, that biology's become reductionist. And I make absolutely no apologies, as Soraia knows, for being a blood and guts pharmacologist, because the only thing that really matters at the end is in vivo, can you give enough safely to get a desired effect whilst minimizing side effects?
Very true. Thank you. Did that answer, Marcelo, your question? Yeah. Okay, more colleagues saying thank you, beautiful work, and Alexander as well, excellent, and our colleague Rosalie Godinho said thank you for the excellent presentation. In the case of PDE inhibitors, many of them can also act as adenosine receptor antagonists. Have you tested whether LFP also inhibits these receptors?
Good question as well. So this goes back, of course, to theophylline, which is a very good antagonist at A2B, A1, and has been suggested to account for a lot of the side effect profile of non-selective phosphodiesterase inhibitors. And you may remember there was Astra developed a drug called N-profilin, which was selective for A2B. It didn't block A1, but it ended up causing cluster headaches and was stopped in its development. But RPL554 does not inhibit any of the adenosine receptors. And we've looked at A1, A2A, A2B, and A3. So again, it seems to be very selective. The only thing that it touched, apart from 3 and 4, as I showed you briefly on the talk, was it does affect PDE10, which is found in the brain. And people have tried to find PDE10 inhibitors for a number of CNS disorders. But again, because it's so bioavailable or poorly bioavailable, it doesn't actually get in a meaningful way into the CNS. So we don't think, although that happens in vitro, that it actually has any translational consequence. But it is a good question. It's something absolutely you need to check, because adenosine receptor antagonists, I think, actually have some merit, but they also come with side effects that you don't want, particularly in people with respiratory disease.
Very good. Is there any more question here? Only compliments, people saying thank you, very good presentation, and thanks for the great presentation. Are there any more questions, colleagues? Any doubt? Any more questions? If there is any other question, is Christina want to close the session, or shall we try to use and abuse of Clive page today here?
Well, thank you for your attention anyway, but you can use and abuse me when I come to Brazil, Soraia. So then I can't escape. But no,
Yes.
please, over to Christina.
Thank you, Clive. Thanks a lot.
Thank you for the great session. Thank you so much, Clive, for this presentation, and Soraia for the moderation of the session. I think it was great because you brought to us both your vision and experience and all the things that you are doing in terms of teaching pharmacology. That is a topic that we are always bringing to these sessions. It's something most of the people in the session, they are from the Department of Pharmacology in different institutions in Brazil. And so I think it's great. Thank you so much. And I think in the future, and especially because you are going to be in our Congress later this year, maybe we can discuss the possibility of joint collaborations and have like an online course that you could contribute to our community, because we also have a group of educational initiatives in our society. And so maybe if Soraia agrees, she is the president of the society now, this is something that we can explore if you agree, because it would be great just to use your experience for our students and graduate students. But thank you so much. And also sharing your research. And so I also would like to thank the audience.
Just a minute, Christina, just to remind Clive that was the type of work that we're trying to bring with Lee Page before he leaves. Yeah, so we are trying to bring this education.
Well, I'm happy, Soraia and Christina, to come. In fact, the first time, Christina, I came to Brazil many years ago was with the British Council, and actually I taught to your postgraduates and some of your graduate students, both in Campinas, Sao Paulo, and also at Fiocruz in Rio. So I haven't been for a long time, so I'd be more than happy to come and help, whether it's online or in person. But because I think the quality of the students that I met then and the number of people I know from Brazil since in the pharmacology community, you have a very high standard already. But if I can help improve it further, I'm more than happy to.
That's great. And so I also would like to thank the audience and register that in this audience we had the presence of two past presidents of our Brazilian Society, Professor Marco Aurelio Martins and Professor Regina Marcos. And so, with that, I would like to say that these sessions that we have been organizing for the last 6 years now, it's an activity that is sponsored by the Brazilian Society of Pharmacology. So that's why it's so great, because the ideas that we have here, the speakers, the colleagues, we all go annually in our Congress and we communicate and bring here topics that we think it's of importance to be discussed and shared with our colleagues and students. So thank you so much. I think it has been great. So I would like to close briefly here. I'll go to these just 3 slides. So just to remind everybody here to that are with us in this session. So we had over 50 participants. We thank all of you for being present. And to remind you that to participate in these sessions, you have 2 options. So you have the one link that is the RCC RNP BR /ao vivo. This is the link that you have to use in case you want to have a certificate to register your attendance for in this session. Or you also have this alternative link that you see in the slide. I would like to remind you that this session has been recorded, and soon, usually like 1 or 2 days, it will be available in our YouTube channel in the ISBEFT web page, where you can also find the videos of all our previous recorded sessions. And so you will be welcome to, we welcome you to go to the YouTube channel and to review this presentation and also enjoy the other sessions and learn from the other speakers. So just to also to remind you, our email is sigfarmaco@gmail.com. And if you have any question or suggestion, please contact us. And so remember to register, and remember to follow all the instructions online if you want your certificate. It should be straightforward. And then just closing by reminding you, our next session will be June 19. Will be the responsible colleague for the SIC Pharmac will be Dr. Paulo Ghedini. Our speaker will be Dr. Gardenia Militão from the Federal University of Pernambuco. The moderator will be Dr. Michele Pereira, also from the Federal University of Pernambuco, and the topic will be lung cancer and modulation by derivatives of N-acyl hydrazones. So we look forward for this next session. So I hope to see you all again next month. And thank you again, Dr. Page. Thank you again, Dr. Costa. And with this, I will ask Emiliano to stop the recording.